Hello guys, welcome to today's video. In this video, I shall be putting you through a topic I call DNA replication. DNA replication. You know, there's something called the Central Dogma of Life. Central Dogma of Life. Central Dogma of Life. This central dogma essentially states that DNA is made from existing DNA, and that from DNA it translates to RNA, and from RNA protein is manufactured. So we have DNA giving rise to DNA. We just call that replication. And then by the way, this is DNA. And then from here, it gives rise to RNA. Okay. And then from RNA, we get something we call protein. So from DNA to DNA is called replication. But from DNA to RNA is called transcription. Transcription. But from RNA to protein is called translation. Okay, so this has been found to be so much true in so many organisms, so many living cells, that it has earned the name a dogma. You know, in science, we really don't believe in dogma. Scientists question everything, and we want to research, we want to understand everything. But this pattern has been found in the overwhelmingly vast majority of living cells across all kinds of species, and hence it has earned the name Central Dogma of Life. So the Central Dogma of Life simply says that replication or DNA is only manufactured or only obtained or only made from previously existing DNA. You don't create new DNA. You create new DNA only from an existing DNA. And then RNA is now made from DNA. DNA now produces RNA, then RNA produces protein. In other words, information of life, all the information, the hereditary information of life that is passed from generation to generation or is used to make cells, multiply cells, or replicate cells. Okay, this is the pattern it follows. That information in DNA is copied by RNA or translated into RNA, and then from RNA, protein is made. Protein is essentially these are the chemicals, or essentially this is the way information in life is expressed in the form of protein. So protein is the final expression that gives rise to the phenotypic characteristics of the animal or plants, as the case may be. But now I said we are looking at DNA replication. So how does DNA replicate or give rise to itself? So now let's go back to our double helix structure. Remember, I told you about this double helix structure in my previous video. So you may want to go check it out where we're talking about the structure of the DNA. Now, let us say we zoom in. We're going to zoom in. Let's zoom and see how this takes place. So if the DNA is going to replicate itself, there are organic chemicals that bring about these processes. Let us say the first we will be looking at something we call the helicase. So I'll be listing those enzymes here, helicase, those organic chemicals. We call them enzymes. They are very vital in the replication process of DNA. So the first, this my double helix structure of the DNA. Remember, it is antiparallel. So if here is 5 prime, this will be 3 prime. So let us say here is 3 prime. Okay, so this is going to be 5 prime. All right, good. Now, these strands, one of them is called the leading strand, another one is called the lagging strand. One is called the heavy strand, another one is called the light strand. So on this heavy strand here, or the leading strand here, replication, the leading strand is replicated in the direction 3' to 5'. That means it is copied in this direction 3' to 5'. So this terminal here will be 5'. This end here, this pole here will be 5'. In the same vein, this other lagging strand here, this end here will be 3'. Okay, so don't forget, existing DNA strand is always copied during the process of replication from the direction 3' to 5'. This automatically means that the new strand of DNA that will be manufactured will always be manufactured or built in the direction 5' to 3'. That is anti-parallel to the existing strand. So let's take it easy. So helicase, an enzyme, comes here and gets here and opens, unwinds. It unwinds this DNA structure. It unwinds this double helix. It unzips it. Unzip. You know, you could view this double helix structure like a zip, and then there's something like a zipper, the zipper of your dress that you pull either up or down. Okay, so this helicase is bringing about the unzipping. It gets into this structure and it unzips it. In other words, it breaks, it cuts these hydrogen bonds that holds the two strands together. So it cuts them open, it separates the strands, it opens up a space, a gap. It opens up a gap in the strand. So if we have something like this, we're gonna have a gap here. Let us say this is what I'm trying to talk about. Okay, so you can just imagine this here. Okay, this is like my double helix structure. Then the helicase comes here and unwinds here and cuts this. There were previously bonds here. There were previously hydrogen bonds here, holding an A to a T, a G to a C. So what this helicase does is it cuts this. It creates an opening, a free space here. Then this space here is called a replication bubble. This is like a replication bubble. Replication bubble. You can see it looks like a bubble, right? So this is a replication bubble. So we have a replication bubble here, you know, close at both ends. But my structure here, I'm zooming in at one of these ends here to give me this structure so we can take a better look at it. That's why I have something like a Y shape here. So this is now called a replication fork. This is a replication fork. This is a replication bubble. So on this end, either end of the replication bubble, you have a replication fork. So this is a replication fork. This is the replication fork is moving this way. All right. So now, after the helicase has done its job of unzipping, the job of the helicase is just to unzip or to open, okay, or to denature or to separate the two strands from themselves, from the hydrogen bonds, to weaken, to cut the hydrogen bonds that holds the two complementary strands of DNA together. So having done that, the next enzyme you're going to have on duty is so let me shift this guys here. I have the helicase. I'll be listing my next thing. We have DNA polymerase, beg your pardon, not so fast, primase. So the next enzyme that will come on duty here is an enzyme we call primase. The primase derives its name from primer, just like helicase derives its name from helix, the helical structure, because it works on the helix to unwind it. The primase derives its name from primer, that because it helps to build a short strand of DNA which we call primer. That primer, technically speaking, is an RNA. Okay, strictly speaking, is an RNA. So it comes here. Let us say at this end, this 3' end here, the base unit here is an adenine and a G and a C. So what the primase is going to do is going to construct a short strand of DNA here, or an RNA here, complementary to this. So in place of a C here, you're going to have a G. In place of a G, you're going to have a C. In place of a T, you're going to have a, in place of an A, you're going to have a T. So this short strand of DNA is what we call the primer. So this is the primer, or better still, let's put this in the box here. So this is what we call the primer. You know, the primer is that short strand of DNA that jumpstarts the replication process. It triggers the replication process. It initiates the replication process, you know, from the word to prime, you know, to prime something, to set it in motion and set it going, set it moving, to kickstart something. So that's like to prime. So the primer is this short strand of DNA that has been made by this enzyme called primase. Don't forget that enzymes derive their name from the kind of job they do, or the substrate they do. So this primer is associated, primase used to manufacture primer, helicase used to unwind the helix structure. All right, so far so good. So we have our primer. Once the primer is there, it becomes the basis for which DNA replication could start. DNA replication proper is carried out by an enzyme called polymerase, or DNA polymerase. DNA polymerase. So this enzyme called DNA polymerase is responsible for polymerization, or for building, or for construction of new strands of DNA. Okay? However, it only acts if there is a primer. It's the presence of the primer that invites it, or that initiates, that causes DNA polymerase to start the process of DNA polymerization. So as soon as there's a primer here, DNA polymerase, we jump on it. So we have DNA polymerase jumps on this and begins to construct a new strand of DNA. This is a new strand of DNA going towards the replication fork. This new strand of DNA will be complementary to the old. So if here is a T, there's going to be an A will be inserted here. If this is a G, a C will be inserted here. If this is a C, a G will be inserted here, and it will be going towards the direction of the fork. Is that understood? So it will be going towards the direction of the fork. So that's the way it will go. So DNA polymerase will replicate this existing DNA in a complementary nature. So the free end of this new strand of DNA here will be 5', whereas the direction that it is going will be 3'. This is 3'. So new strands of DNA will always be manufactured in the 5' to 3', in a complementary fashion to the existing DNA, which is 3' to 5'. So this is on the leading strand. By the way, this is leading strand. Don't forget, leading strand. So on the leading strand, this will keep going while the helicase keeps opening this strand, unwinding this strand. So the replication fork will be heading this way. This is the direction of the replication fork. It will keep unzipping and going this way, while the replication of the leading strand will be coming behind it, just following it up. Okay? That is replication on the leading strand, which is pretty straightforward and clear. But on the lagging strand, there is a little bit challenge. That's because the lagging strand, you can see the orientation. 5' is on this end. Meanwhile, the replication fork is going this way. But remember, I just said that the rule of thumb is that new DNA is being replicated, or new DNA is being formed or manufactured always in the 5' to 3' direction. Whereas existing DNA is always copied, as a rule of thumb, from 5 prime to 3 prime. But you can see from this sketch that the 3 prime is this way and the 5 prime is that way. So the only direction that we expect that the new strand of DNA will be copied or will be made has to be from my left to my right, this way, because this is 3 prime for the existing DNA and 5 prime for the existing DNA. So this process will be repeated. Primates, we always hop on board here and create a short strand of DNA we call the primer. That primer, we copy it outward this way, but let's put it over here, because as at then, this fork was yet to open this wide. We just imagine that the fork started opening gradually, this way, okay, as it is moving this way. So time was when it just opened up a little bit like this. So primase creates this enzyme primer. So this is a primer again. All right. So this was copied from 3 prime to 5 prime. So went this way. It's the end of the strand, and that is all. Okay, complementary. Complementary meaning A versus T, T versus A, G against C, C against G. Just in that way. You understand me? All right. So complementary copy. So the new strand is created this way. So for this new strand, this will be the 5 prime end of it, and this will be the 3 prime end of it. Complementary to this. So as the fork opens further, more space is created inside. So as more space is created towards the fork, another little chunk of DNA called the primer will still be created again. All right. So by the primase. So when the primase creates this, DNA polymerase comes on board and construct this new DNA until it hits the first primer. So on the lagging strand, this is called the lagging strand because this is where the replication starts. The replication here is lagging. It's coming from behind. This one needs the replication fork to open to some level, all right, to some appreciable level before the primase could do its job. So once the primase constructs the primer, DNA polymerase gets into action and constructs a new DNA complementary, new DNA strand complementary to the existing strand, complementary to this lagging strand, and then it constructs it complementary, and then it constructs it from the direction it copies this existing one from the direction 3 prime to 5 prime. In other words, constructing a new strand of DNA that will be orienting from 5 prime to 3 prime, anti-parallel to this. So when that is done, and then the fork opens further inwards or further backwards, if you like, then another DNA short strand of DNA primer will still be constructed again, and then DNA polymerase comes on board and replicates this strand from this primer until it hits the next primer. So we are going to be having on this leading strand one continuous strand of new DNA. There will be a continuous strand of old DNA and a continuous strand of new DNA. However, on the lagging strand, we're going to have a continuous strand of the existing DNA, but the new DNA will be in piecemeals. It will be in pieces. So you're always going to have primer strand of DNA, primer strand of DNA. As you go backward again, you're going to have primer strand of DNA, primer strand of DNA. That's the way it will continue. So there will be pieces of DNA. These pieces of DNA on the lagging strand, these fragments of DNA is what we call Okazaki fragments, named after the scientists that discovered them. So Okazaki fragments. All right. So Okazaki fragments will consist of primer DNA strand, primer DNA fragment, primer, DNA fragment, primer. All right. So after the DNA polymerase, then we will have a ligase. But even before that, we have exonuclease, which is just an enzyme that will come up and digest this primer. Exonuclease will come here and digest these primers, remove them up, remove them up, remove them up, and some few nucleotides will be fixed there that will be complementary. And then at the end of the day, we're going to have the last enzyme here, ligase. Ligase will now join them up, join this up to the next one, join it up, so that on this ligase strand too, you will end up having a continuous strand. So ligase does the job of ligating or joining the Okazaki fragments together so that they become also continuous. So in summary, we just said that two strands here, leading and lagging. So the difference is that on the leading strand, there's a continuous replication, okay? While on the lagging strand, there's a discontinuous replication. And that's because of the orientation of the lagging strand, okay? So from 3' to 5', is going from away from the replication fork. So a chunk of DNA, a little primer is created, and then copying replication is done outward away from the fork. Then inward again, another strand of another primer needs to be manufactured by primase again. So primase gets the primer, then DNA polymerase builds the new strand of DNA, and then exonuclease comes in and removes those little primers that are in between the DNA fragments, because those primers, like I said, technically speaking, they are RNAs and they cannot be part of the DNA. So they are removed. They are removed by this enzyme called exonuclease, and then afterwards, another enzyme called ligase now joins them together. After all of this is done, so these components here will separate, and these components here will separate. So here you are going to have a new strand versus an old strand. Over here too, you're going to have new strand and an old strand. Okay, you're going to have, so in each of these two helices, there will be a new strand and an old strand. Here too, there will be a new strand and an old strand, and they will be arranged or oriented in anti-parallel fashion. This will be 5', 3'. Here will be 5', 3', and so on, 3', 5'. And here too, we're going to have a 5', 3'. Okay, so based on this, we always say that the replication of DNA is semi-conservative. Semi-conservative because an old strand is always preserved, and then a new strand is attached to it or replicated or formed to join it. An old strand always bonds or pairs with a new strand. So it is semi-conservative. We don't have two new strands together or two old strands together. We always have in each of these helix structure, one old strand and a new strand, one old strand and a new strand. So basically, this is the structure of DNA. This is the DNA, the way DNA is replicated from one cell to another, or even within the cell. I will see you in my next video. Like and subscribe. See you.